Now we will move to our next topic, which is the multilayered QC in CGT from vector fidelity to epigenic regulation. And I would like to welcome for this topic William Mauck. He's a team leader and molecular biologist from the Solvius. That, William.
Thank you.
I can give you 5 minutes.
Okay. Thank you. All right. Thank you very much for the introduction, and it's really great to be here. I just came up from Raleigh, North Carolina, where we're opening our Center of Excellence in Cell and Gene Therapy and Large Molecule. And so I kind of want to start this talk off talking about some of the applications of cell and gene therapy. And on the right, we have a young girl who actually has metachromatic dystrophy. And so she has been treated with a cell therapy where they took the stem cells and they inserted the correct gene, functional gene into there, and then inserted them back into the patient and have cured her from this disease as well. And this was just a couple years ago. And we have a slightly longer study of Emily Whitehead, who had a CAR T-cell treatment, and where her T-cells were taken, created the CAR cells and CAR T-cells, and then introduced into her body. And most recently, as of just, I think, earlier this year, there's a lentiviral treatment that's an inhalation treatment where they're actually the patients are inhaling the lentiviral particles, and it's going into the epithelial cells to treat cystic fibrosis and modifying that gene. So that allows for both cell and gene sort of therapy treatments out there. And these are just really great examples, and there's plenty more of them as well. However, while all of these examples exist, it's really important that we recognize how long and how much time it takes. And I think our previous two talks have emphasized the length of time it can take to have any of these assays testing be done, and how it impacts the treatment options for patients to deliver these therapeutic products to them to help treat their diseases. And so there's a lot of various steps in that manufacturing process that occur. And within that, when we're talking about cell and gene therapy, we have make sure that gene of interest or that construct is still intact. We want to understand where it's at. Making sure there aren't any deleterious impacts of the placement of that into the genome, as well as vector constructs is intact and there aren't essentially duplication events or losses of parts of that construct that could have an ultimate impact on how effective that treatment is, as well as understanding that the viral, if there's any viral contaminants in there, and ensuring that those don't get passed along if they were inadvertently introduced into the manufacturing process to the patient. And so this emphasizes just the generality of a production cycle and where each of these could happen at different points, and then you want to test for stability. As earlier talks had said, when we're dealing with master cell banks, working cell banks, we need to ensure that same therapeutic product gene of interest that's inserted into those genomes is producing the same product consistently over time between different lots or batches. And so the way we do this at Solvius for characterizing the construct is we utilize this method of targeted locus amplification, or TLA, and next generation sequencing, which in general, you take dissociated cells and you fix them so that all the DNA is kind of locked and cross-linked in place. It goes through an enzymatic fragmentation so that the portions of the DNA are more manageable, and those portions that are in proximity closer to each other are then ligated back together, creating these circular DNA fragments that can be amplified. And so you have your construct or your gene of interest, and now it's associated with can easily be amplified with the surrounding area around that gene of where that insert occurred. And this allows us to characterize and understand if we have the integration sites, if we have duplication events, co-integrations of multiple inserts, indels that occurred in that construct, rearrangements, concatenators, and the only thing that kind of rounds out the whole characterization here is the vector copy number, which we have a parallel ddPCR assay that we run alongside the NGS that allows us to easily quantify how many copies of each insert occur in a cell. And so if we start looking at the higher level of this, the data, then we are looking at a heterogeneous T cell in this example. And so these are all the insertion breakpoint sites that you would have. And so this isn't just happening in a single cell, but across like a whole population of cells that were isolated. And so within this, if you were to, you can get an understanding of like where all those insertion breakpoints are. You can expand those cells out, and you can start delving into where if there are any deletion events, inversions, translocations or duplication events, all characterizing how effective is this construct going to be in producing the reliable and producing the therapeutic product that you intend, or how, if the cell itself is that therapeutic product, how well it's actually going to function when you give that back to a patient. And so, as I said before, this includes stability studies, so we're going to check this over time. So you're looking for that same pattern that you're going to find if these cells were expanded and frozen down into master cell banks that were utilized over time. And so it's easy to also view this in a tabular report, and so you can kind of get a really quick picture of where all of these breakpoints happen and understand, like, are these occurring in the gene in the intronic regions and the exon regions of the genome? Are they occurring upstream or in non-coding regions of the genome? And it allows you to quickly understand if they're on the antisense or sense side of the genome. And it really allows that delving into seeing a little bit more carefully, we can look at where in the genome these are occurring and categorize where these insertion breakpoints are and where these genes are, the constructs are being inserted. And we find that almost 50% of them are occurring in the intronic regions. Very few, only 5% are found in the within exons, and only 8% of them are found in front of or upstream from any known genes. And so this is extremely important so that we ensure that the insertion points don't occur near any proto-oncogenes that may inadvertently induce tumor growth and uncontrolled cell growth, which would very much not be the goal of what this therapeutic product should be. As well as, and this all tracks in with our lentiviral viral mechanism, because lentivirus inserts itself in areas of open chromatin where there's active transcription happening. So this is normally happening, we'd expect, within genes in that intronic regions or where there's open chromatin. So this sort of a pattern is as expected. And diving a little even deeper into it, we can look at the area right around the construct and ensure that the gene of interest and the associated proviral sequences are all intact. We use multiple priming sites to ensure that if for some reason the priming site of one seems to have a mutation in it, that we don't have that loss, we can cover that area. And it gives us confidence that that construct itself is as intended and what we wanted to see, and ensuring that there's no mutations or frame shifts, indels that are occurring there that could cause complications in that gene. And so all of this is characterizing our insertion, our genomic insertion of our target locus or our gene of interest. And so we want to ensure the highest safety regards for that product as a therapeutic for patients. And we also want to ensure, and I'm going to pivot here to looking at viral safety and utilizing next generation sequencing, because we want to ensure that safety of the product is also doesn't contain any adventitious viral agents that may be passed from the production, the manufacturing process into the patient's sample. And so as an overview, we have for the NGS process, we isolate the total nucleic acid, so RNA and DNA. The RNA is reverse transcribed into cDNA, and then that, along with the DNA, is processed through library preparation and then hybridized to over 1 million long sequence probes that are utilized to cover over 2,500 different species or viral variants out there. and then we can test for. And so these all encompass a broad range, so it's trying to capture as many different variants that exist for different virus strains. And this allows us to have a very broad yet sort of targeted approach towards looking for common viruses or viruses that may be introduced into the manufacturing process. And so once they're sequenced after that probe hybridization, we sequence them on the next generation sequencer and analyze that data with our own internally developed pipeline that allows us to align any of the sequences we have against what our expected viral, all the different variants of virus species or strains that we have in our database. And this isn't required to be an all-encompassing. It's possible to have either a taxon specific, so you have a rodent panel or a human panel or a bovine panel, depending on what your product was grown in, and you might want to be more specific to like what species of cells that gene therapy is developed in. Or it could be that full panel of over 2,500 different viral products or viral strains. And in each one of these, we always measure this as our cutoff or our threshold we were talking about, kind of that in the previous talk was, you don't want to set your parameters too stringently or too low so that you accept everything. So I guess the Goldilocks is right in the middle at 50%. And so where our cutoff is, if we have 50% of that genome, then we would count 50% or more, we count that as a positive evidence of that virus being in that sample. And so this allows us to detect, have some sort of a cutoff and threshold. And I'll give an example here in a minute. And this ensures that we're actively detecting the virus, because we also have that system suitability control, or we call it spike in positive control, in every sample that goes in there from the very beginning. And so this is a known sequence that's part of that database, and we should be seeing it at a constant quantifiable amount, because we know how much we put in the beginning there. So we have that in every sample that we run, as well as negative controls. And so looking at the data here, we can kind of go over these graphs. It's really easy. The first two, the minute virus of mice and the human coronavirus strain, both of those are positive. So we found those would be positively found in our assays, because we have over 50% of the genome detected. Now, there's different intensity or levels that may have potentially be due to a different level of contamination or expression for how much virus is in the sample itself. And so you can see that on the chart, there's different intensity levels across the genome that are relative to the expression or the amount, but this isn't quantifiable directly. But we do have, in the case for the murine leukemia virus, we do have 2 large spikes. However, the total is much less. We don't nearly meet 50% of that genome. And so this would be negative to any viral contamination in that sample. And these large spikes may be leftover portions of a viral genome that have just been carried through and have amplified in the NGS library prep process. And so this would still be a negative because there's no viral genome to actually replicate itself in there. And real advantages of utilizing a next-generation sequencing are the speed and the consistency that you can perform this on different types of samples. And so where you can use that on cell-based samples, we can use that on cell-free samples like the lentiviral therapeutic that the cystic fibrosis patients were receiving in the example earlier. And so this can work on any of the cell or gene therapy kind of products out there. And it's fast. It has really low input, so you don't need a lot to test multiple different species consistently, so it can work on really small volumes. It's regulatorily accepted. It's becoming more and more prevalent. It's a recent change to the regulatory guidelines. And so we're working with that, and it's helping to deliver the product, the therapeutic, to the patients quicker and allowing them to feel confident that the product they're getting is safe to take. And that's really what we want to see to help patients get better. And so I'm going to take this time now to kind of shift gears to talk about epigenetics. And now, the epigenetic field is like incredibly large, and it's blowing up, and there is a ton of material out there. There's a ton of testing that's being done to understand all the different roles in the way epigenetics can regulate cells and the cell products that are being made. And so I'm just going to dive into a couple here, and I think it's really important to understand that this is a really burgeoning field, and how we're kind of emphasizing this this one kind of paper we're looking at is they're utilizing the epigenetic regulators to improve hematopoietic stem cells in their lentiviral gene therapy. And so what they found was the hematopoietic stem cells are, when you get them, they're really hard to maintain. They're really hard to like culture long term. They want to differentiate, and then they're not going to stay in that like multipotent stage. And so along with that is once you transduce them with lentivirus, then even more of them kind of die off or differentiate, and it's even harder to expand that population and expand those cells. And however, when they added quinostat and CPI-203, they found that it enhanced the stem cell or the hematopoietic stem cell expansion. It preserved their function or their non-differentiated state, and improve their lentiviral transduction. So this all really seemed like kind of a no-brainer here when understanding like this seems to be an improvement all around. They took CD34 positive cells at day one. They cultured them for 4 days in media with their standard STF media that had cytokines in it, and then they had two conditions where they added each of these drug products. and measured and found that the hematopoietic stem cells had frequencies of live cells much greater than the controls, and the proportion of the hematopoietic stem cells was much greater. So these maintained longer in culture their hematopoietic state and didn't differentiate, and they are allowed for increased amounts of construct to be inserted into the genome there. And so this really makes sense because the quinostat itself is actually, interferes with the deacetylation process. And so it's essentially keeping all of the areas of the chromatin kind of, or the histones kind of open and accessible, which is your stem cell state, is like they're all like ready to just either like shut down or like differentiate based on which parts of the genome kind of get turned on and off. But if they're all on, they're all in that like stem cell, like haven't differentiated into differentiated yet. And so this seems to make sense. However, it's important to recognize that because of that like increased lentiviral vector transduce, then you have to be careful, because now if you're dealing with if you're thinking you have the same infection, like transducing them with the same amount, it's going to be very much you can't use that same amount. So this is something that would need to be taken into account very early on in the phase of drug development, so that you're qualifying and validating the media that you're growing these cells in and maintaining them, and ensuring that you have all of that information properly documented so that you can do your vector copy number. There's not an extensive amount of additional insertions that are happening that could cause either concatenators or inversions or anything else that's happening. And so that really pertains to not just that vector copy number, but like where that's happening, going back to characterizing where in the genome this is now occurring, because if a lot more of that chromatin is open and accessible, now you have insertions maybe in a higher likelihood of happening in your proto-oncogene spaces, and that definitely wouldn't be something you wanted to introduce in this case. So there's additional testing that needs to be taken into account in understanding that process, as well as residual particles of the epigenetic regulator therapy. And so this could be carried on and through and needs to be tested to ensure that the the therapy itself, whether it's the cells or some sort of a product, doesn't also contain any of those epigenetic residuals. And so it's, I think, kind of tying this back all together and a little bit with Solvius in RTP is we have cell-based bioassay team that does potency, that has flow cytometry, and FACS sorting. We have an analytical chemistry team that has mass spec and UPLC, HPLC, so we can quantify the size and shape of lentiviral vector, the charge on that, as well as the molecular biology team that I'm in charge of, and we're expanding our automation capabilities. We're growing in as far as the different assays that we can perform and our experience with epigenetics, as well as single cell, which was brought up earlier as transcriptomics and multiomics, so that you can combine not just the transcriptome, but you can look at the proteome, you can look at the epigenetic markers as well. And so there's lots of avenues to explore this and lots of ways that tying this all together into the most robust honed product, ensuring that you have consistency and stability for those products so that you can deliver this to patients quicker and safer is really what we're aiming for and how we're going to do that utilizing next generation sequencing technology. So just wanted to call out Solvius there and thank everybody for coming.
So how do you balance the depth of the analytical data with turnaround time and cost in a multi-layered QC framework?
Well, the turnaround time is quicker because you're not relying necessarily. Are you talking about in general, or are you talking about
in general, like you are doing the multi-layer QC.
Mm-hmm.
So how that you talk about the NGS, then you talk about the different tools. So how that turnaround time, because like mass spec assay, that's take time. Mm-hmm. Even that if you want to go a little bit in a single cell RNA-seq, that takes time. Yeah. So that's my question there, that if you have how you balance in a QC environment, that turnaround time with the multi-layer assays.
So because we have it all in-house, I'd say, it really makes it easy for us, at least in RTP and in our global sites as well. They have a really tight network, so we're able to either do it all in the same place so that we can be processing cells and growing those cells, as well as then taking parts of that and running either for mass spec or for single cell transcriptomics, say, or just adventitious viral testing, all kind of in line with it right at the same time. So I'd say that's probably the quickest way that we increase that turnaround time, as well as just adding as much automation in different steps as possible, and really helps ensure we can meet those turnaround times and do that quickly.
Very good. Any other question for William? If not, let's give the big round of applause for our all three.
Thank you.